All right, I'll start. Hi, I'm Jacques. I'm a bio major. We're the Golgi bodies, and we're going to be presenting on chapter one.
I'm Luke. I'm a Kines major, and uh, yeah, we're doing chapter one.
I'm Jacob. I'm a biology major, and yeah, we're just gonna go over a couple sections in chapter one.
And I'm Garrett Ryman, and I'm a biochem major.
Alrighty, I'm going to start us off for this session. I'm also going to be keeping track of our concept map. So, starting off, we're going to start with section 1.1, which talks about microorganisms. They're also known as microbes, and they're life forms that are too small to be seen by the human eye. Um, they're a very diverse group. They inhabit every environment that is able to support life. Some of them are single-celled, some are very complex structures, and some are multicellular. And they tend to live in microbial communities, which is a community that just exists as like a large group of microorganisms in one location. A good example of that would be the depths of Michigan lakes that have various different communities that live there. Um, Microorganisms are also the oldest form of life. They are a major portion of Earth's biomass. They surround plants and animals and affect human life in both positive and negative ways. There's many infectious diseases. They aid with our food and water, soils, animal health, and our fuel. Um, there's many tools that are needed to study microbiology and studying our microorganisms. We use microscopes, and in order to do that, we need to have cultures, which are cells that are grown on a medium. Medium is a liquid or solid mixture that contains all the required materials and nutrients for growth. Growth is the increase of a cell number that results from cell division, and colonies are the visible. It's visible to the eye, and they contain millions or billions of cells. Um, yes.
Okay, so the next section is the structure and activities of microbial cells. So the cell is a living compartment that interacts with the environment and other cells. And the elements of this structure are uh, so what all cells have in common is the cytoplasmic membrane, which is a barrier that separates the inside of the cell from the outside of the environment. The cytoplasm, which is an aqueous mixture of macromolecules, small organics, ions, and ribosomes inside the cell. Ribosomes are protein synthesizing structures, and then the cell wall is present in some microbes, and it confers structural strength. Um, then next is prokaryotic versus eukaryotic cells. This is just a structural division. So prokaryotes includes bacteria and archaea. They have no organelles, which is a membrane enclosed structure, and also no nucleus. Eukaryotes is the third domain of life. It includes plants, animals, algae, protozoa, and fungi. These contain organelles. They have a DNA enclosed in a membrane-bound nucleus, and they have mitochondria and chloroplasts. Next is talking about genes, genomes, nucleus, and nucleoid. So a genome is a cell's full set of genes. In eukaryotic DNA, they have linear chromosomes within a nucleus. They're much larger and they have more DNA. In prokaryotic DNA, it's typically single circular chromosome that aggregates to form the nucleoid region. They may also have plasmids, which is extra chromosomal DNA that confers special properties, and they are small and compact. And then the activities of microbial cells. In nature, cells typically live in microbial communities. For metabolism, it's a chemical transformation of nutrients. Enzymes, which is a protein catalyst, transcription, DNA information converted to RNA. Translation is RNA used by ribosome to synthesize protein. And for DNA replication, it's just copying the genome. And lastly, for the activities of microbial cells, they can also be motile, which is when cells move through self-propulsion. Differentiation, which is some microbes that modify structures to form specialized cells. Intercellular communities, which is some microbes respond to chemical signals from other microbes. And lastly, evolution, which is genetic changes transferring to offspring over time.
The next section is all about cell size and morphology. And morphology just means like the size and shape in relation to each other for the microbe. And most microbes are super small. In fact, like for prokaryotes, there are only zero point 0.2, like the smallest prokaryotes are 0.2 micrometers, but they can get up to 600 plus micrometers in diameter. Whereas eukaryotic cells are normally 5 to 100 micrometers in length. Prokaryotes are normally between 2 and 0.5 and 10 micrometers. So on average, prokaryotic um, microorganisms are smaller. For the surface to volume ratios, there are many advantages to being small. You have more surface area in relation to your cell volume. And the smaller you are, the relatively larger your surface area is in relation to your volume. Which this means that high diffusion rates and efficiencies are encouraged, like just through their body type. And most bacteria import things via diffusion or other forms of molecular transport. So they support greater nutrient and waste product exchange per like square inch or square micrometer of like actual cell. So they're just more efficient the smaller they are. And there's also formulas to calculate exactly how efficient something theoretically is, and that's the surface to volume ratio, which is just surface divided by volume, and this gives you the relative size of the surface area in relation to the volume, and also therefore shows relatively how efficient a cell is at diffusion. And then other morphological uh, categories that come into play is the like different types of cells. So a coccus cell, for example, would be spherical. A rod or bacillus would be cylindrical. A spirillum is a flexible spiral, while a spirochete is a rigid spiral. And then there's also appendage bacteria, irregular or asymmetrical bacteria, and then some just say grouped and clustered together after cell division. But these clusters can form specific shapes that are categorized, such as diplococci and streptococci, among others. And there are many variations to the basic morphological types that we know about.
So, Introduction to Microbial Life, section 1.4. There's three main domains or lineages: bacteria, archaea, and eukarya. Bacteria includes only prokaryotes and differentiate single cells from 0.5 to 10 micrometers long, but they vary widely. Archaea is also prokaryotes, and they have 5 well-described phyla. They're usually associated to be extremophiles, meaning that they live in extreme environments, which it could include high temperatures or low temperatures or low nutrient levels. They don't have parasites or pathogens for plants and animals, and they have 12 phyla instead of 5. And then eukarya is the multicell organisms that we're more used to, like plants and animals and fungi. Originally, they were cellular, but many have appeared 2 billion years ago. There is 6 kingdoms instead of phyla, and they vary dramatically in size, shape, and physiology. Viruses are another microbe. They are obligate parasites that only replicate within a host cell. So it's sort of debated on whether or not they're truly living organisms on their own because they need a host. Um, so they're not cells and they don't carry out metabolism. They simply just take over infected cells or infect those cells rather and rely on those metabolic processes to live and reproduce. They have small genomes, double-stranded or single-stranded DNA or RNA, and classified based on structure, genome composition, and host specificity, such as bacteriophages.
All right, before we hop into 1.5, I just want to go over so far what we've got on our concept map. We're starting with microorganisms, dividing it into 4 different subplots, including prokaryotes, eukaryotes, viruses, and also having one subplot being the structure in all cells. So the major plot points that we're looking for are the key components to these different groups. So when we go from prokaryotes, we're splitting into bacteria and archaea, then going down into morphologies and the actual structure of prokaryotes. We do the same with the eukaryotes. We talk about the viruses and we're able to describe how they're different than all these than cells due to the fact that they are not cells themselves. And then we also wanted to clarify the structure that is present in all cells. All right, continuing with section 1.5, we're going to go into the microorganisms and the biosphere. The history of life on Earth is very old. Earth is 4.6 billion years old, and the first cells are said to have appeared between 3.8 and 4.3 billion years ago. At the time, the atmosphere was anoxic, which means that there was no oxygen present. And that was up until that way. It was 2.6 billion years ago until that changed. So there were only anaerobic metabolisms. The first anoxygenic phototrophs appeared 3.6 billion years ago. And then cyanobacteria, which are the oxygenic phototrophs, first appeared 2.6 billion years ago. And then the most recent development would be plants and animals, which are estimated to have appeared around 5.5 billion years ago.
So this just goes over the summary of how life came to be on Earth during that timeline. And this means that bacteria, archaea, and eukarya are all distinguished. And it also means that everything is descended from at least one last universal common ancestor. Speaking of microbes, there are 10 to the 30th power. There are believed to be 10 to the 30th power microbial cells on Earth. And the total carbon is a significant fraction of Earth's biomass. Extremophiles, such as archaea, live in habitats that are too harsh for other life forms. So these are hot springs, glaciers, or areas with high acidity. Microbial ecology is how microbes affect plants, animals, and the entire global ecosystem. And there's some charts showing how microbes for nitrogen and phosphorus are the majority suppliers, even more so than plants, out of all the life on Earth. Um,
So the impact that microorganisms have on human society can be both beneficial and harmful, such as disease or fertilizers for agriculture and food production, as well as other valuable human products such as medicine, energy generation, or environmental cleanup. Um, Microorganisms as disease agents, we've been able to control infections with antibiotics and stuff like that. Although most of the time we're talking about viruses and bacteria. I feel like it's in a negative way, but most microorganisms are actually beneficial in terms of, like we've been talking, food preservation, wastewater treatment, and vaccinations and antibiotics. Many aspects of agriculture depend on microbial activities, such as nitrogen-fixing bacteria and cellulose-degrading microbes. You also have a vast gut biome that helps digest carbohydrates and things along those lines, helps synthesize vitamins and other nutrients within your body. An example of nitrogen-fixing bacteria can be looked at in soybeans. The root nodules contain the bacteria which can fix nitrogen, which is very important to the plant growth. And so the nitrogen can come from that bacteria through ammonia, through waste, and through other things such as fertilizers. So of course there is negative impacts with food in particular. It can cause spoilage as well as disease. So there's a lot that goes into safety and prevention of spoilage that these microbes can induce. But at the same time, there are positive impacts with preservation. There's also the production of dairy products using yeast and things like that, as well as other food products, which can be preserved, like sauerkraut and pickles. Alcohol fermentation can produce the alcohols that we see today and can drink.
So lastly, microorganisms are used in industrial or just technology standards. So industrial microbiology is using microorganisms for major industries such as pharmaceuticals and brewing. And then for biotechnology, they can be genetically engineered microbes making high value products in small amounts for some of the beneficial microbes. And it has a huge impact on human industry. So microbes can be used to produce valuable products, biofuels. They can be used to clean up our waste. They also have major impacts on industry because biofilms can clog and corrode pipelines and holding tanks in factories, ships, and even the oil industry. So for the production of biofuels like methane and ethanol, wastewater treatment, bioremediation, which is cleaning up pollutants, and biofilms, which is just the growth on submerged surfaces like pipes, drains, storage tanks, implanted medical devices.
All right, so that about brings the end of where we're going to be covering today, but I would like to once again go over the new additions that we made to our chart. In addition to what we've just been talking about, we have to realize that there are many benefits and negatives to microorganisms as we covered. So as Garrett said perfectly, we always view all these things as negative, but we fail to realize how many benefits come from microorganisms in our lives. So it's important that we realize that the pros of a lot of these microorganisms like greatly outweigh the cons of it. Now, is there anything else that y'all would like to add to this conversation before we wrap it up?
Uh, make sure we show the paper.
Yeah. So I know it's going to be inverted for y'all, but this is where we're at right now. As I said, we take our microorganisms and we divert them into different quadrants. We additionally added the impacts of human society to our chart. We were able to get a more solid definition of extremophiles, and we're able to also see that not all extremophiles are archaea as previously thought in the studies. Um, yeah, so we were able to get a good bit done, and we plan on continuing to work on this a little bit further. But at this point, have a very solid concept map that's coming up. Luke, did you also have something you wanted to say?
No, I didn't have anything else to add.
Okay,
All
I'm
right.
all good here too.
all right.
Does it?